Okay, so hello everyone, my name is Vasundhra and today we are discussing the most expected questions of plant protection, which includes plant pathology and entomology. So good evening everyone. So let's start with the first question. Which forecasting model is specifically developed for predicting fire blight in apple and pear? What is it? Good evening everyone. Now, this is the first question for you on your screen, that which of the following forecasting model is specifically designed for predicting fire blight of apple and pear? C? Is it C? Its name itself indicating mill stable, mire blight, blast AM or epi-pre. So these models name are basically derived from the organism or the disease itself. Okay. So here we can clearly see that very good, mire blight is the correct answer. Mire blight is for what? Fire blight. What is the causal organism of fire blight? The causal organism of, very good, mire blight is the correct answer. Causal organism for fire blight is Arvenia. What is it? Arvenia amylovora. Is it clear? Okay. Arvenia amylovora, very good. So the forecasting model is mire blight. What about mill stable? Mill stable, that is used for predicting. What is it for? Anyone? Venturaria inequalis. Venturaria inequalis. And what is the disease that is caused by Venturaria inequalis? That is apple scab. Is it clear? Now, what about this mire blight we have discussed, caused by Arvenia amylovora? That is fire blight of apple and pear. Is it clear? What about blast AM? Is there any disease called as blast? Anyone? Any disease which is present in a crop? Blast disease? Blast of rice. Clear? Very good. Rice blast. So blast AM is used for predicting the what? This blast of rice, and it is caused by, very good, that is Pyricularia oryzae. Is it clear? What about epi-pre? Epi-pre is not a forecasting model, but it is a decision support system. Decision support system. It is also used to predict or identify the disease, but it has capability for decision making, and it is used to identify the disease of wheat. To detect the disease or to predict the disease of wheat. Is it clear? So what will be the answer? So in your last exam also there were questions from this forecasting model. So you have to remember all the forecasting model that is related with the different disease. Is it clear? So these are the different forecasting model, decision support system that are related with different pathogen. Okay. So let's move to the next one. Next we have, which of the following oomycetes caused black pod disease of cocoa? Black pod disease is caused by which of the following? Is it Phytophthora infestans, Phytophthora palmivora, Phytophthora colocasia, or Phytophthora nicotianae? Their name indicating. Very good. Phytophthora palmivora is the correct answer. So most of your questions is from disease part. So we will discuss the different causal organism and cover each and every one. Now tell me what is the disease caused by this Phytophthora infestans, and tell me about the importance of this Phytophthora infestans. Firstly, tell me what is the disease caused by Phytophthora infestans. Very good. It is causing disease in potato, that is late blight of potato. Very good. And it is related to what? Late blight of potato is related to what? Anyone? Late blight of potato, what is it? Irish famine. Very good. In which year? Irish famine occur in which year? What is the year? You have to remember the year. Very good. Irish famine in the year 1845. Very good. Okay. Next we have Phytophthora palmivora. As we know, it causes black pod disease of cocoa, and symptom as its name indicating, the cocoa pods turn black. Okay. Phytophthora palmivora. What about Phytophthora colocasia? Colocasia is the related with what? Taro. So it is causing taro leaf blight. Is it clear? Okay, what about Phytophthora nicotianae? Nicotianae is associated with tobacco, so causing blight in which crop? Tobacco. So this will be common, that they will give you different similar options for a question, okay, and they will ask one of them, like black pod disease of cocoa. So all the option appear similar. Okay, so you have to carefully pick the right one. Next we have now virus transmission. Again, this is one of the important topic for your exam. Okay, which virus is transmitted by aphid Myzus persicki in non-persistent manner? Specifically non-persistent manner. Although they are asking similar, like this much of question. But here I specifically design it for specific one, like non-persistent manner. Okay, potato virus. Why? Very good. This is the correct answer. And what is this non-persistent manner? So we have what? Non-persistent, semi-persistent, and persistent. So what is happening in non-persistent? So in non-persistent, very good, this is a stylet bond. It is present only in the stylet of the insect. That is, the virus is present only in the stylet of the insect. And when it feed on a healthy plant and transmit virus to that healthy plant, then it do not retain the virus. Okay, that is, there is no latent period in this. Virus is not persisting in the body of the vector. Okay, that is non-persistent. Semi-persistent, virus is present in its vector for some time. Okay, after that he has to feed again to acquire that virus. And what about persistent? In persistent, it is a circulative type. It is circulating in the whole system of its vector. Is it clear? So here, what is it? Non-persistent, vector do not persist virus. Okay, vector do not persist virus in it. Okay, it is also called as stylet bond. Is it clear? Semi-persistent, it can hold insect vector, or it can hold the virus for some time. And what about this persistent? The virus is present in the virus persists in its vector. There is another term Trans-ovarial transmission. Okay, what is it? In this, the vector, what vector is doing? It is passing this virus to its next generation through eggs. That is trans-ovarial transmission. Is it clear? Okay, so among them, non-persistent type is potato virus Y to its progeny. Very good. What about this potato leaf roll virus? This is also what? This is also transmitted by aphid, that is Myzus persicki, but in persistent manner. Is it clear? Both are transmitted by Myzus persicki, but this potato virus Y do not remain along with the vector, but in case of potato leaf roll virus, it persists with its vector. That is a persistent one. Next we have tomato spotted wilt virus. Okay, so there are two diseases in tomato. We will discuss them. One is tomato spotted wilt, or we can write it as, and next is tomato leaf curl virus. Okay, so it is transmitted by thrips. And what about leaf curl? It is transmitted by white fly. This is again very important question that they can ask, that which of the virus or which of the virus is transmitted by white fly and which one is transmitted by thrips. So it should be clear that leaf curl virus is transmitted by white fly and tomato spotted wilt virus is transmitted by thrips. Is it clear? What about this rice tungro bacilliform virus? So rice tungro bacilliform virus is transmitted by leaf hopper. Is it clear? So it is transmitted by leaf hopper. So there will be a question from this part, as in your previous question paper, many of the question is from this part. Is it clear? Clear or not? So I have written here also that potato virus X is transmitted by this several other aphid in non-persistent manner, and also tomato spotted wilt virus transmitted persistently and propagatedly by thrips. Again, one more important thing about rice tungro bacilliform virus is it is associated with green leaf hopper and it require rice tungro spherical virus. This is again very important for vector transmission. Okay, so it requires its spherical virus for its transmission. This is also very important. Is it clear? Nephotetics viruses, very good. This is the name of the leaf hopper. Very good. So you have to read this topic, that what is the transmission method or how the viruses are transmitting, what are the viruses that are transmitted by aphids, what are the viruses that are transmitted by the white fly. So this topic is very important in exam point of view. Is it clear? Okay, next again from the disease part, which bacterial disease of rice is caused by Xanthomonas oryzicola? Again, this is very important and confusing one. Yes, whether it will be bacterial leaf blight or leaf streak, what will be the answer? Leaf streak, yes, leaf streak. There is a very minor difference between the pathogen of leaf blight and leaf streak. What is the pathogen of bacterial leaf blight? What is it? Xanthomonas oryzae. Pathobar oryzae. Okay, so here answer will be bacterial leaf streak. Okay. Is it clear? Again, this question, in your last exam, the question was aflatoxin is produced by which of the following? That was the question in your last exam, and the answer will be Aspergillus flavus. Again, there is another question I have framed here. Which of the following fungi produce phemonosins? Which of the following? Aspergillus flavus. I just told you that Aspergillus flavus produced aflatoxin. What is the toxin? What is it? Verticilloid? Yes, very good. Fusarium verticilloid is the correct answer. Phemonosin is a, what is it? Toxin that is produced by Fusarium verticilloid. Okay? And what about this Aspergillus flavus? The toxin produced by Aspergillus flavus is aflatoxin. Is it clear? What about Penicillium expansum? What is the toxin produced by Penicillium expansum? What is it? That is patulin. Patulin is the toxin that is produced by Penicillium expansum. Very good. Next is Fusarium verticilloids. We have discussed. It produces phemonosins. And next we have Claviceps purpurea. It produces argot alkaloid. Is it clear? So again, this portion is very much important, that what are the toxins produced by the different fungus. Next we have, which of the following fungi belong to the order Tephrinales? And what is the specific thing that is present in the member of this Tephrinales? That's why it is important. What is it? A, very good, Tephrina deformans. Now, why Tephrina deformans is important? What is important in this? What kind of ascus it is producing? It is producing any ascocarp or not? That's why it is important. What kind of ascocarp is present in Tephrina deformans? Anyone? Okay, so Tephrina deformans is important because it do not produce any ascocarp. It produces naked asci. Is it clear? That's why it is important. It do not produce any ascocarp. And these are the disease that is produced by Tephrina deformans. One more important thing is it is causing leaf curl. Generally, leaf curl symptom is associated with viruses. This is very important. Generally, leaf curl symptom is associated with viruses. Generally, but here, this is the fungus that is causing leaf curl in peach. Tephrina deformans is this fungus that is causing leaf curl. So that's why it is important that name the fungus that is causing what? Leaf curl in peach. Or they can ask, peach leaf curl is caused by which of the pathogen? Fungus, bacteria, virus, viroid. Okay, so answer will be fungus. Is it clear? Claviceps purpurea, argot of rye and other cereals. This is also very important, that what is the argot fungus we can also say. Okay, so Claviceps purpurea is the fungus that is producing argot and causing argotism. Is it clear? Next we have Astilagomydes. It is a common smut of maize, and one of the important thing is it is an edible delicious Okay, this is edible. So they can ask which of the following smut is edible. So you have to remember Astilagomydes is the edible smut, and the edible product of its of this fungus is Utila cocae. This is very important. They can ask that which of the following is an edible smut. So Astilagomydes is the edible smut. Is it clear? Smut is internally or externally? Both. Some of the smut are internally born, some are externally born. We have questions on this, internal or externally born. Yes, loose smut is internally seed born. Very good. Okay, next is Venturi inequalis causing apple scab. Okay, and the important thing here to remember is what is the resting structure of the apple scab or this Venturia inequalis, that is pseudothecia. This is also very important. They can ask what is the resting structure for apple scab. This is pseudothecia. Is it clear? So these are the important things that are related to all these options. Okay. Next, we have the bacterium associated with Stuart wilt of maize. Which bacterium is associated? Answer is very clear. Pantoia stewarti, subspecies stewarti, is the bacteria that is associated with. So again, this is a different disease like Stuart wilt of maize. So they can directly ask what is its causal organism, what is the causal organism of this disease, or they can give you causal organism. They can ask that it is causing which of the following disease. Okay. Next, we have other options: Pseudomonas syringae, Pathobar syringae, and it is causing which disease? That is bacterial blast and canker in several crops. Again, disease of the crops is important topic for your exam. Most of the question were asked from this topic. Okay, so Pseudomonas syringae causing bacterial blast or canker of several crops. So what is the important symptom for bacterial disease? What is the initial symptom for the bacterial disease? This is also very important. So the initial symptom of bacterial disease is water-soaked lesion. Again, this is very important. What is the initial symptom of bacterial disease? That is water-soaked lesions. Is it clear? Yes, very good. Water-soaked lesion is the correct answer. Now, next we have Arvinia amylovora, that is fire blight of apple and pear. And one of the characteristic symptom of fire blight of apple and pear is shepherd crook. This is again very important, that shepherd's crook is the typical symptom for fire blight of apple and pear. You have to remember this one, that if they ask shepherd crook is the characteristic symptom of which of the following disease, so it is present in, okay, so it is present in Arvinia amylovora. Next we have Pantoia stewarti, as we have discussed. So it is causing Stuart wilt in the maize. And I have written here also what is its characteristic symptom, that is long pale green yellow longitudinal streaks on the leaves. This is the characteristic symptom for this Pantoia stewarti. Is it clear? Next is Xanthomonas campestris, Pathobar campestris. Again, there is a characteristic symptom for this disease and you have to remember it. And this is the V-shaped lesions. V-shaped lesions are the characteristic symptom of Xanthomonas campestris, Pathobar campestris, causing black rot of crucifer. Okay. So these are the typical symptoms for different disease. Shepherd crook is for fire blight of apple and pear. V-shaped lesion is the characteristic symptom of black rot of crucifers. Is it clear? And what about Pantoya stewardii? What is its typical symptom? That is pale green to yellow longitudinal streaks on the leaves. Generally, streak symptom is also of the virus affecting leaves. Okay. But in Pantoya stewardii, there is also yellow longitudinal streaks present. Is it clear? Again, you have to remember shepherd crook, you have to remember V-shaped lesions, and this long pale green to yellow longitudinal streaks. Is it clear? And what is the first symptom of bacterial disease? That is water-soaked lesion. This is also very important. Is it clear? Now, next we have, which of the following disease is transmitted by polymyxabete? Now you will tell me what is this polymyxabete? What is it? Answer you already know that what is the answer, but what is this polymyxa? Obviously, it is a virus transmission vector, but what is it? Is it bacteria? Is it virus? Is it fungus? What is it? Yes, bacteria is correct, but what is it? This is not bacteria. Polymyxa is a fungus. Okay. So this fungus is transmitting this rhizomania of sugar beet. So there are different fungus that are transmitting the viruses. Like polymyxa we have. Next we have this Spongospora subterranea. One of the important question is again that what are the fungus genus, or what are the different fungus that are virus vector? So answer will be polymyxa. Next we have Spongospora. So these are the two important fungus you have to remember. Is it clear? Yes, fungus is the correct answer. So what about Rhizomania sugar beet? We discussed polymyxa betae is the fungus that is transmitting this virus. Okay. That is wheat necrotic yellow vein virus. Next we have wheat spindle streak mosaic virus. So wheat spindle mosaic virus is again transmitted by polymyxa species but graminis, polymyxa graminis. Genus polymyxa but species is graminis for wheat spindle streak mosaic. What about this potato mop top? Again, this is important and very common viral disease that can be asked that potato mop top disease is transmitted by which of the following? So again, it is transmitted by vector is Spongospora subterranea. You have to remember this. Potato mop top is important virus. Is it clear? Next we have tobacco rattle viruses. Tobacco rattle viruses, again this is also important because tobacco rattle virus is also transmitted by nematode. Okay? Trichodorus and paratrichodorus. This is very important. Again, that which of the following nematode species are are helpful or are causing virus transmission? So answer will be Trichodorus and paratrichodorus. Again, this is very important one. Trichodorus and paratrichodorus, polymyxa and Spongospora, that is fungus and these are the important nematodes that are transmitting the different viruses. Is it clear? Next, we have Biotech mixture was first developed for the management of which of the following? And who discovered this Biotech mixture? Again, this is from the important topic, that is epidemics. PMA Miladit is the correct answer. Very good. Now tell me among which of the following disease? B. Very good. B is the correct answer. Downy mildew of grapes. Okay. PMA Miladit discovered what? Biotech mixture. When he was accidentally discovered, Biotech mixture, when he was working with the downy mildew of grapes. That is, what is the causal organism of downy mildew of this grapes? What is the causal organism? Viticola plasmopara. This is plasmopara viticola. Is it clear? So, PMA Miladit discovered Biotech mixture when he was working with downy mildew of grapes. Again, I have written here also, Biotech mixture was developed by PMA Miladit in 1885. This is again very important, in Bordeaux, France. And it is the first successful fungicide used in the plant disease management. If we have to prepare 1% of Biotech mixture, then we will use, what is its composition? It contains copper sulfate, quicklime and water. So if we have to prepare 1% of Biotech mixture, so we are taking 1 kg copper sulfate, 1 kg quicklime and 100 liters of water. Is it clear? 1:1:100 ratio. Clear? It's not 1:100. Is it clear? Again, one more symptom is important in apple scab, that olive green velvety spots on leaves. This is very important. Again, important symptom in apple scab, that olive green velvety symptom is present on both leaves and fruits. So you have to remember this also. Clear? Next, we have, which of the following fungi produce chlamydospore as an important survival structure? This is very common. Chlamydospore is present in which of the following? Resting structure. Fusarium, very good. Fusarium oxysporum is the correct answer. Most of the fungi, generally, they are overwintering or they are producing only mycelium as their resting structure. Okay, in most of the fungus, mycelium is the resting structure. But in case of some of the species that are producing dormant spores, like chlamydospores, we have, so it is a resting structure of Fusarium species. So what is the disease caused by the Alternaria solani? Again, this is very important to discuss here. So early blight of tomato and potato is caused by Alternaria solani. Fusarium oxysporum, most of the Fusarium wilt. Okay, most of the Fusarium wilt is caused by Fusarium oxysporum. So Fusarium is important pathogen that is associated with the wilt in various vegetable crops. Is it clear? And Cercospora viticola, that is causing Cercospora leaf spot of sugar beet. Now its name indicating that it is causing disease in beetroot, sugar beet. Next, we have Coltotrichum gliosporides that is associated with the anthracnose disease. Always, Coltotrichum is associated with the anthracnose disease in many fruits. Okay. So, again, if we talk about the Alternaria solanae resting structure, as I mentioned that most of the fungi, they are resting in their mycelium. That mycelium is their resting structures. Okay, in case of Alternaria, mycelium and conidia are the resting structure. Next, we have Fusarium, we have discussed. Chlamydospore is the resting structure. Next, we have Cercospora. Again, mycelium. As I discussed, mycelium is the resting structure for Cercospora also. And what about Coltotrichum? Acerbulae is the resting structure for what? Coltotrichum gliosporide. And now Acerbulae is sexual or asexual structure? Anyone? Is it sexual or asexual? What is it? Asexual. Very good. This is asexual structure and it produces conidia. And what is the shape of this structure? Acerbulus is which kind of structure? Anyone? This is saucer shaped. Is it clear? Now tell me different asexual structures. What are different asexual structure? Acerbulae is one of them. Very good. Now tell me other. Anyone? Pycnidium. Very good. Pycnidium is one of them. Pycnidium is again asexual structure. What about other? Sporodochium. Very good. Sporodochium. So these are the asexual structure. And what about sexual structures? What are the sexual structures? Anyone? Cinema. Very good. Cinema is one of the asexual structure. Now tell me what are the sexual structures that is present in the Ascomycota. What are they? Apothecium, very good. So this is also very important that different asexual structures. So different asexual structures are Pycnidium, Sporodochium, Cinema and this Acerbulae. They are the asexual structure. And if we talk about the sexual structure, what are those? Clistothecium, Perithecium, Apothecium, Gymnothecium, okay, and Pseudothecium. So again, this is very important that they can directly ask that which of the following structure is asexual and which of the following structure is sexual. Clear? Next, we have which of the following fungi is the causal organism of loose smut of barley? Which of the following? Its name indicating. Barley. It's very good. This is Astilago hordi. Sorry, loose smut. I am asking loose. Yes, loose smut. This is not. This is covered smut of barley. And this nuda is loose smut of barley. Okay, what about Astilago tritici? Loose smut of wheat. And what is this Tilicia caris? What is it? Okay, so this is bunt of wheat. Okay, Tilicia caris is bunt of wheat. Now, loose smut of wheat, what is its characteristics? The main characteristics is black powdery mass of spore is present in entire ear. Entire ear is covered with this black powdery mass of spores, which are teleospores. Okay, what are they? Teleospores. Is it clear? And this Astilagotriticide is, what is it? Internally seed borne. Is it clear? This is internally seed borne. What about this Astilagohordii? This is covered smut of barley, and the important characteristics is smut sori remain covered with a silvery membrane. This is the important symptom for this disease, and this disease is externally seed borne. This is externally seed borne disease. Is it clear? What about Astilago nuda? That is loose smut of barley. Again, symptom will be same, that black powdery spore mass, that is of teleospores, that cover its entire ear. Okay? And it is, what is Astilago nuda is internally seed borne. This is internally seed borne disease. Clear? Next we have Telecia caris. This is common bunt of wheat. Okay. So, yes, there is a fishy odor, that is trimethylamine, that is associated with Telecia. Okay. It is associated with Telecia. And it is externally seed borne. So this is again very important. These are the 4 different diseases that we have discussed here, and you have to remember whether it is internally seed borne, whether it is externally seed borne, internally seed borne or externally. Which one is internally, which one is externally. So among them, this loose smut of both barley and wheat. Okay, loose smut of barley and wheat, that are internally seed borne. And what about covered smut of barley? That is externally seed borne. What about Telecia? That is also externally seed borne. Is it clear? Anyone that was asking about the seed borne? Now it is clear which of the following is internally seed borne, externally seed borne. Is it clear now? Okay. So you have to remember this, and also these symptoms, that where is the silvery membrane present, where is this black powdery mass of spore is present. Okay, and this trimethylamine. Yes, in case of like in Karnal bunt also, there is a fishy odor, that is due to trimethylamine. This is very important one. That is a fishy odor. You have to remember this also. This is also very important. Okay? Recorded class? It is available on YouTube, no? Next we have, which of the following disease is transmitted by Olpidium brassicae? Again, this is, what is this Olpidium brassicae? This is also fungus that is transmitting the viruses. Okay? So, which of the following? Anyone? So, lettuce big vein disease is the correct answer. I have written the answer here. You are still not answering. So we will discuss about the vector. Again, this lettuce big vein disease, this is again very important disease. Lettuce big vein disease is transmitted by again a fungal genera, that is Olpidium brassicae. Previously we have discussed the two genera, that is, anyone? What are the other two fungus that we have discussed that are responsible for transmission of viruses in last question? What were they? Spongospora and what was other one? Polymyxa. What about rice stripe virus? That is transmitted by small brown plant hopper. That is Lodelophex striate, Spongospora, Polymyxa, Olpidium. Yes, Spongospora, Polymyxa, Olpidium is the correct answer. Next we have Barley yellow dwarf disease. That is by the aphids. Citrus Trestija viruses. This is again very important. Citrus Trestija viruses. You have to remember the causal organism of Citrus Trestija viruses, and causal organism is aphid. This is important. Or this is brown citrus aphid. That is Toxoptra citricida. Again, this is very important that they can directly ask. Citrus Trestija is the most common disease and it is transmitted by Citrus Trestija viruses. And what is the vector for transmission of this virus is brown citrus aphid, which is Toxoptera citricida. Okay. So let's move to the next one. Which scientist first established the heterogeneous nature of wheat stem rust? This is very important, and if you have attended the classes of AO AAO, we have discussed this particular question or this history part in that. Anton de Very, very good. This is the correct answer. Who is Anton de Very? Father of plant pathology. And this is the important contribution of this, that heterogeneous nature of rust fungi. What is heterogeneous nature of rust fungi? That rust is present in 5 different, or rust produces 5 different spores. What are they? Pycnospore, Achiospore, Uridio, Teleospores and Basidiospores. And these two are present in which? Primary host or secondary host? Anyone? Yes, very good. Pycnospore, Achiospore, Uridospore. Now tell me, these two, Pycnospore and Achiospore, are present in wheat or barberry? And what is alternate host? Barberry, very good. This is present in the barberry and it is a what? Secondary host or alternate host, we can say. Is it clear? Question can we ask, what is the alternate host of wheat stem rust? So it is barberry and it is present in wheat. At this stage, basidiospore, which is the haploid spore. Again, this is very important. This is the haploid or sexual spore of rust. Haploid sexual spore. Okay, and it is present in the soil. Is it clear? Clear about the rust? What is its alternate or secondary host? What are the different spore stages that are present in the rust? So, this concept was given by the Anton de Very. Next, we have Erickson. What is the contribution of Erickson? The three are related with the this wheat stem rust. Okay, so what is the contribution of Erickson? So Erickson studied about wheat stem rust, that rust follows the Mendelian fashion. This is also very important, that these rust follow all the Mendel's law. Okay, so that was discovered by Erickson, that this rust follow the Mendelian fashion. Next is also important, Stakesman. What is the contribution of Stakesman? He gave the physiological races. And what are they? Simply we can say that these are the morphologically similar. Like we can talk about a wheat stem rust. So all these species will be same, but they differ in their pathogenicity. Like suppose we have Puccinia graminis forma specialis triticale. It is affecting one variety of wheat, okay, but it is not affecting other variety of the wheat. So the other one that is affecting this variety, so these both are the physiological races. Is it clear? That suppose we have two varieties, okay, and both are prone to wheat stem rust, but this Puccinia graminis triticale, that is affecting this particular this one variety, and other race is affecting other variety of wheat. So these both are physiological races. So this concept was given by E.C. Stakesman. What about K.C. Mehta? This is related to Indian pathology. K.C. Mehta, anyone? Contribution of K.C. Mehta. Father of stem rust in India. Yes, very good. He studied the stem rust in India, or father of stem rust in India. Very good. Is it clear? All these points are important. Okay, one more question is important in Euridiospore and Teleospore. And what is that? Euridiospores are the repeating spores. What are they? Repeating spores. And what about teleospores? These are the resting spores. Is it clear? Repeating spores, or we can say summer spores, and resting spore, or we can say winter spores. Is it clear? Summer and winter also. This is also important. These are the summer and repeating spore, and teleospore are the winter and the resting spores. Is it clear? Okay, so let's move to the next one. The active ingredient metalaxyl belongs to which frac group? Anyone? Group 4. Yes, we will understand what is it. So what is FRAC? FRAC is Fungicide Resistance Action Committee. Okay. So FRAC is Fungicide Resistance Action Committee, and it classified different fungicide into the different group. Okay, so here we are discussing group 1, group 4, 11 and group M03. So here you have to remember what? Sometimes this can be asked. This is mode of action. Okay, so what comes in the group 1? These are the carbamates. Here important is carbamates. Okay, carbamates come under group 1. What about group 4? Phenylamide. They can directly ask, they will not ask like which of the following come under the FRAC, but they can ask their mode of action because these are simple one. Okay, so what is the mode of action of carbamates? Different carbamates. And also you have to remember the example of this fungicide group. That what are the example of carbamates? Carbendazim, venomil, thiophanate, methyl. From here this question is more important, that these fungicide belong to which group, which fungicide group. This you have to remember. Next we have phenylamide. What comes in the phenylamide? That is metalaxyl, M, mifenaxam or benalaxyl. So they can directly ask that metalaxyl comes from which fungicide group. In your last exam, carbendazim was asked that carbendazim belongs to which group. So you have to remember the fungicide and their group. Is it clear? Next we have QOI, or we can call them strobilurins. Okay, these are the fungal derived fungicides. Okay, they are derived from the fungus. Okay, so examples are azoxystrobin. They can directly ask that azoxystrobin belong to which fungal group. So again, this is very important topic. You have to remember fungicidal group and example of the fungicidal group. Okay, next is dithiocarbamates. Dithiocarbamates include mancozeb, this is important, manef, this is important, zinef, this is important. And here in strobilurins, azoxystrobin is very important, and in phenylamides, metalaxyl is important, and in carbamates, carbendazim is important. That is asked in your last exam. Is it clear what you have to study from this topic? Next we have, which of the following fungal genera produce muriform conidia? Now what is muriform conidia? Muriform conidia, what is muriform? What is the meaning of muriform? Anyone? What is muriform conidia? These are the different questions. So there are chances that they can ask, like what is muriform conidia? Yes, shape, what kind of shape it have? So muriform conidia contain both longitudinal and transverse septa. Is it clear? So it is typically present in what? Alternaria. Okay, alternaria. And stem phylum is also one of the example of fungus containing muriform conidia. Is it clear? Very good. B is the correct answer. And what about fusarium? Anyone? What is the shape of conidia present in the fusarium? There are two types of conidia present in the fusarium. Anyone? Fusarium? What type of conidia they are producing? Fusarium? Anyone? Sickle shape. Very good. Sickle shape conidia. But which one? Is it macro or micro? There are two types of conidia present in fusarium, that is microconidia and macroconidia. Okay. So micro and macroconidia. So these macroconidia are very good sickle shaped. And what about microconidia? These are spherical shaped. Okay, so rod shaped, yes. Is it clear? Micro and macroconidia. What about aspergillus and penicillium? Anyone? What is the type of conidiophore structure they are producing? What is specific in both aspergillus and penicillium? What is it? So the conidiophore structure of this penicillium is paintbrush-like. Paintbrush structure. This is very important and this is generally considered Is it clear? Because this is not widely open. In case of Penicillium, it is not widely open. But here in Aspergillus, it is properly widely open. That's why it is called as toilet brush structure, and it is called as the paint brush structure. Is it clear? So you have to remember the shapes from here. What is muriform conidia? Where both transverse and longitudinal septations are present. What about fusarium? Micro and macroconidia are present, and both Aspergillus and Penicillium we have discussed here. Clear? Here I have also written that brush-like. So we have discussed it that muriform. Yes, one other thing, muriform conidia is also called as dictyospore. Okay, other name is dictyospore. Okay, next we have which fungicide primarily inhibit mitochondrial respiration by blocking electron transfer at QOI site of cytochrome B. Again, this question is from fungicide. So again, they can ask their mode of action. So mode of action, fungicide group, this is important. And fungicide group, you have to remember for different fungicides. So I have written answer here, azoxystrobin is the correct answer. Next, here I have written their FRAC group. They are not asking this you in the exam, but I have written that they come under which of the FRAC group. And next, I have written the mode of action. We have discussed carbendazim. What is its mode of action? Inhibit beta tubulin formation. Azoxystrobin, mancozeb, captan. At least you should know the mode of action for important fungicides. So these are the basic one. Carbendazim, azoxystrobin, mancozeb, captan. You have to remember their mode of action. Okay, some of selective or some of important fungicides. Next is causal organism of kernel bunt of wheat. Anyone? Yes, this is Talicia indica. Causal organism of the kernel bunt of the wheat. They can directly ask you that what is partial bunt, what is stinking bunt. Okay, so Talicia indica is kernel bunt of wheat. Again, I have written here which of the following is internally seed borne and externally seed borne. So most of the bunt are externally seed borne. Okay, these are externally seed borne. And what about this Astilago triticide? We have studied about this loose smut of wheat. And one question we have learned that Astilagomydes is the common smut which is edible. Is it clear? Common smut which is edible, that is Astilagomydes. This is also important. Clear? And again, they can directly ask you, name the causal organism of flag smut of wheat. You have to remember this is Urocystis agropyri. This is again important, that what is the causal organism of flag smut of the wheat. This is Urocystis agropyri. You have to remember the causal organism for this disease. And again, kernel bunt of the wheat, that is Talicia indica. Common bunt, that is Talicia caris. Okay, so these two are important one, that which one is common and which is kernel bunt, and also called as stinking bunt. Stinking bunt is Talicia caris and this is partial bunt. Next we have, which of the following bacteria is the causal organism of bacterial wilt of solanaceous crops? If we talk about the fungus, we know that Fusarium is associated with the wilt disease in most of the crop. And if we talk about which bacteria is associated with the bacterial wilt, so it is Ralstonia solanacearum. Okay, Ralstonia solanacearum, very good, C is the correct answer, is typically associated with the bacterial wilt. Next we have Arvinia amylovora. Fire blight of apple and pear, we have discussed an important thing. What was here? What was the important thing? What is the important symptom for the fire blight of apple and pear? We have discussed. Again and again I am telling you, this is shepherd crook symptom. This is the typical symptom for fire blight of apple and pear. Next we have black rot of crucifer. You have to remember causal organism, that is shepherd crook. Yes, very good. Xanthomonas campestris, Patho campestris. Next again Ralstonia solanacearum. This is causing the bacterial wilt of the tomato. And Pseudomonas syringae, Patho tomato, bacterial speck of the tomato. You have to learn all the disease and their causal organism. In your previous exam, directly they are asking disease and its causal organism, or they are giving you causal organism, or you have to identify that the disease they are causing. Is it clear? Again, which one of the following is an example of systemic acquired resistance inducer? Again, this is very important. This is because this is specific one. Tomato virus. Which tomato virus? We have two types of tomato viruses we have discussed, that is tomato spotted wilt virus and other one is tomato leaf curl viruses. So it is transmitted by thrips and it is transmitted by what? Whitefly. So these are the two tomato viruses that we have discussed. Is it clear? Okay, so this is important, that because this is the example of, typical example of systemic acquired resistance inducer. What is systemic acquired resistance? It is a type of resistance. Okay, so this is inducing resistance in the plant so that it can resist from the pathogen attack. Okay, so acylbenzolar S-methyl, you have to remember this as it is, that acylbenzolar S-methyl is an example that is inducing the systemic acquired resistance in the plants. Is it clear? Next we have, which of the following is a causal organism of downy mildew of pearl millet? Downy mildew of pearl millet. A. Pearl millet. Parasitica. This is Sclerospora graminicola. Okay, so what are the different diseases that are caused by these organisms? First one is Perennospora parasitica, that is causing downy mildew in crucifers. Parasitica causing downy mildew in crucifer. What about Sclerospora graminicola? That is causing downy mildew in pearl millet. Is it clear? Plasmopara viticola, we know downy mildew of grapevine, and other is Bremia lactuca. As its name indicating, it is causing disease in lettuce. So these are the different causal organisms of downy mildew. They can directly ask you the causal organism from here. Again, citrus canker is caused by. What is the causal organism of citrus canker? A, very good. This is Xanthomonas citriae, subspecies citriae. This is the causal organism of citrus canker. So, other one, Pseudomonas syringae, Patho syringae. Again, I am telling you, this is very important. You have to remember the causal organism. Bacterial blast or canker, we have discussed it in the last question also. And Agrobacterium tumefaciens, this is again very important, that crown gall disease is caused by Agrobacterium tumefaciens. Is it clear? Next we have, which of the following pathogens survive in the soil mainly as oospore? Oospore. Now you will tell me oospore is a sexual spore or asexual spore, and it is produced by which of the phylum or which of the organisms are producing oospore, and it is resting spore or what? B, Phytophthora. Yes, Phytophthora is the correct answer. Now tell me, oospore is sexual or asexual spore? What is it? Oospore is a sexual or asexual spore? B is the correct answer. What will be the answer here? Oospore is sexual spore that is producing by Oomycota. And what are the members of Oomycota? Pythium, Phytophthora. They are producing the what? Oospores. Is it clear? Now we will discuss, you will tell me what are the different sexual spores. Again, this is very important question. What are the sexual spores? Different sexual spores. Anyone? So, oospore is one of them. What is other? That is produced by the Oomycota, Pythium and Phytophthora. Okay. Next we have zygospores. This is also sexual spore. Zygospore are produced by Zygomycota. And what are the member of Zygomycota? What are the member? Zygomycota. Oospore, Basidiospore, Ascospore. Very good. Zygomycota, Mucor and Rhizopus. Next is Ascospores, Basidiospores. These are the sexual spores. This is sexual spore. And what about the asexual spore? What are the major asexual spore that we know? First is Chlamydospore. These are the asexual spore. Okay. Conidia. This is also asexual spore. Next we have zoospores. These all are asexual spores. Is it clear? This is very important, that which of the following is sexual spore, which of the following are asexual spore. Okay, yes, Chlamydospore is sexual. Now again, we have two terms, that is pycnium and pycnidium. Pycnium and pycnidium. Now you will tell me which of the which among two is sexual and which is asexual. Sporangiospore, yes, sporangiospore is very good. Sporangiospore is also a sexual spore. Now these are the two terms, pycnium and pycnidium. What are the difference between both pycnium and pycnidium? Which of the following is zoospore is a sexual spore? Which of the following is, which of them is asexual and which is sexual? Pycnidium, fruiting body. Yes, pycnidium is a fruiting body. What about pycnium? It is also a structure. Now, in case of pycnidium, it is a asexual fruiting body. Yes, correct. And what about pycnium? This is a sexual body that is present in rust. So again, this is very important that they can give you like which of the following is asexual. They will give you pycnidium, pycnium, sporodochium, acebuli. And they will ask which of the following is sexual. So pycnium will be the answer. There are two terms, pycnium and pycnidium. Yes, pycnidium is asexual fruiting body, but pycnium is a sexual structure that is present in the rust. Okay, that is producing pycniospore in the rust. That pycniospore we have discussed. So pycniospore is produced in this pycnium. Is it clear now? This pycnium is producing pycniospores. Clear? Okay. So I have written here major survival structure. We have oospore is also the survival structures. Astilagomyides, they are also surviving in mycelium and conidia. We have discussed, and alternaria is also surviving as the asexual or mycelium structures. Okay, Puccinia graminis, we all know. Yes, this is also very important. We have discussed this also, that teleospore are the resting spores, or we can say them winter spores in case of rust and smut. Clear? Next, we have teleomorph of Coltotrichum gliosporides belong to the genus. And firstly, tell me what is teleomorph and what is anamorph. What is anamorph and what is teleomorph? Anyone? What are they? Yes, glomerella is the correct answer. This is correct. But tell me what is teleomorph and what is anamorph. What is this teleomorph? This is again important topic. You have to remember the teleomorph and anamorph of different fungus. Okay. Now tell me what is anamorph. The asexual stage. That fungi have two stages. That is sexual and asexual stages. So the asexual stage is called as anamorph and sexual stage that is called as teleomorph. Is it clear? Very good. Asexual is anamorphic and sexual is teleomorphic. Again, this is an important question. They can ask that anamorph is asexual, sexual, resting, or none of these. So you have to remember anamorph is a asexual stage and teleomorph is a sexual stage. Correct? You have to remember all the anamorph and telomorph of different fungus. So what is the anamorph of, what is the telomorph of Coltotrichum gliosporides? That is glomerella. Singulata. Very good. Glomerella is the correct answer. Correct? Next, we have which one of the following is an obligate parasite. Firstly, you will tell me what is this obligate parasite. Obligate. What is the meaning of obligate parasite? Anyone? Obligate parasite means they require living host to complete its life cycle. Is it clear? Living host is required to complete its, yes, very good, to complete its life cycle. So among them, there is Albugo candida. Albugo candida is the one that require the, and it comes under which? And what is the disease caused by Albugo candida? Again, this is very important. Very good. White rust. Good. White rust of crucifer. This is again important disease. That white rust of crucifer is caused by Albugo candida and it is a member of Oomycota, lower fungi. Okay, white rust of crucifers, that is caused by Albugo candida. That's why it is important. Okay, and it is an obligate parasite. Again, this is important. Name the organism that is causing the or that caused the white rust of crucifers. So it is Albugo candida. This is very important. So I have written here also. Okay, so these are the anamorph slide is here. Coltotrichum anamorph part. This is anamorph and telomorph. So telomorph of Coltotrichum, that is glomerella. Fusarium telomorph, gibberella. Now tell me, what is this gibberella? Any disease associated with gibberella? Important disease associated with gibberella. What is it? Quickly. Disease associated with gibberella. Gibberella fusicori, yes. What is the disease? Organism is Gibberella fusicori. But what is the disease? Seedling foolish, yes. Or we can say Bacne disease. Very good. Again, this is very important question, that Bacne disease is caused by which of the following? That is Gibberella fusicori, which is a telomorph of what? Fusarium. Okay. And again, take all disease of wheat. The disease with unique name you have to remember. One more disease is a take all disease of wheat. What is the causal organism of take all disease of wheat? That is Guamanomyces tritici. This is also important, these two. Bacne disease and other one is take all disease of wheat. Causal organism of take all disease is Guamanomyces tritici. Is it clear? Or graminis. Okay. Guamanomyces graminis. Clear? So these two are important, Bacne disease and wheat take all disease. Next we have, which of the following disease is effectively managed by development of multi-line varieties? Gibberella fusicori. Again, I am telling you, these are their just anamorphic and telomorphic stages. Okay. So if Gibberella fusicori, Fusarium fusicori, they are just their telomorph and the anamorph. Is it clear? So let's move to the next one. So next we have, which of the following diseases effectively managed by development of multi-line? B, yes, this is again important, stem rust of wheat. So how we are managing stem rust of wheat and why multi-lines are important? Firstly, we will understand, yes, multi-line are the mixture of pure line, that is resistance is wide. Okay, so it contain, these are the mixture of genetically similar line that differ only in their major resistance gene. Okay, so there are different genes present in them. Since this airborne pathogen, okay, stem rust is airborne pathogen, okay, which follow a particular path that come from south to north India. Okay, so it is a airborne. So we are using multi-line varieties in case of stem rust of wheat. This is also very important question, that because stem rust of wheat is the specific example of this, that where we are using the multi-line. Is it clear? Next, we have questions. Which insect is commonly known as red hairy caterpillar of groundnut? A, very good, Amsecta albistriga is commonly known as red hairy caterpillar. Okay. And what about other? Spodoptera litura. This is the Common name of this is tobacco caterpillar. Helicoverpa armizera, gram pod American wallworm. They will directly again, they can directly ask you the question of the pest and their common name. They have given you common name, they will give you common name and will ask what is its scientific name. Okay, Helicoverpa, very dangerous. Okay, so it is a gram pod borer. Okay, it attacks many crops. Yeah, this is very dangerous. Okay, next is Agrotis epsilon, that is black cutworm. Again, one important question is there. These Amsecta albistriga is major red hairy caterpillar that is present in South India. And next we have Amsecta murray, that is the red hairy caterpillar that is prominent in the North India. Is it clear? This is important here. Clear? So this is important that Amsecta, this albistriga is major in South India and murray is common in the North India. Clear? And here you have to remember one more thing, that is this black cutworm. What is the typical symptom for a cutworm? Cutworm is the larva cut seedling near the ground level. This is important, near the ground level. Okay, and usually night time. Next we have, which of the following predator is widely used in biological control of aphids? So here majorly they will ask that questions from parasitoid, predators. Okay, that what is the predator of that, this one is the predator of which of the following they will ask. Okay, so answer will be Chrysoperla. Okay, Chrysoperla, that is lace wing. What about this Trichogramma chilonis? It is an egg parasitoid and it is used against Lepidoptera pests such as Helicoverpa, Spodoptera and stem borers. So from this part you have to remember what are the major parasitoid, what are the major predators, okay, that are attacking this insect and pest. Next we have Chrysoperla. Chrysoperla is a green lacewing and it is affecting aphid, whitefly, jessid, thrips and mealybugs and mites. Next we have what? This is Bracon habiter. This is larval parasitoid. Okay, Bracon habiter is a larval parasitoid and Encarsia is also a parasitoid. And these are the different target pests in which these all parasitoid and predator are acting. So in our question we have asked about the predator. Is it clear? Next we have, which of the following insecticide belong to diamide group? Again and again I am telling you, you have to remember all the insecticide name, insecticide or pesticide name and their group. So here it belongs to the chlorantraniliprole. Okay, so you have to, very good, this is the correct answer. So you have to remember chemical group like chloropyrifos, it belongs to organophosphate and their mode of action is not that much important, like they may not ask this, but you just go through from this PDF that I have written here. And what about imidacloprid, which chemical group it is belonging to? That is neonicotinoid. Okay, they can directly ask that imidacloprid belong to which of the following group. Next is this hypermethrin, synthetic pyrethroid. This is synthetic pyrethroid. So you have to remember insecticide, pesticide and their groups. Is it clear? Next we have this, what is important from this insecticide, other thing, that is this chlorantraniliprole is the first commercial successful anthralinic diamide. Is it clear? This is the first successful or first commercially successful insecticide. Again, this is important. Okay. And one more important thing, you have to remember the trade name and chemical name of pesticide and insecticide. Is it clear? Which of the following insect exhibit hypermetamorphosis? Which of the following? Hypermetamorphosis. Which of the following? Meloe species, Helicoverpa armizera, Bombyx mori, or Didercus singulatus. Which among the following? Anyone? This is Meloe species. What is Meloe? That is blister beetle. Okay, what is this blister beetle? Now, what is hypermetamorphosis? There are different instars that are present in the larval stage. Okay. Next, we have complete metamorphosis. Complete metamorphosis means all the stages are present. Egg, larva, pupa, adult that are present in it. Okay. Helicoverpa armizera having the complete metamorphosis. What about Bombyx mori? It also has complete metamorphosis, which we call holometabola. And next we have Didercus singulatus, that is red cotton bug. But this red cotton bug has incomplete metamorphosis. Okay, what we call it? Hemimetabola. That is incomplete metamorphosis. That is pupal stage is absent. They only contain egg, nymph and larva. Egg, nymph and adult. Is it clear? What is hypermetamorphosis? That is different stages are present. Different larval instars are present. Okay. And what about this complete metamorphosis? Egg, larva, pupa, and adult. That is all the stages are present. And incomplete pupal stage is absent. Next is which parasitoid is extensively used against the sugarcane internode borer? Again, you have to remember parasitoid, predator, insecticide part. Okay. So, which parasitoid is extensively used against sugarcane internode borer? I have written answer here, Cotacia flavips. Okay. So, I have written all the parasitoid here and their common name also. So, first one is Trichogramma japonicum. What is it? Egg parasitoid wasp. That is, and its type is egg parasitoid as its name indicating, and it is effective against rice stem borer, sugarcane borers, okay, and Lepidoptera eggs. Clear? And what about Cotacia flavips? It is also a brachianoid parasitoid eggs. Okay, wasp, and it is a larval parasitoid. It is a larval parasitoid that is effective against sugarcane internode borer. Clear? Next we have Bracon habiter. This is again the larval parasitoid and it is effective against Helicoverpa, Corycera, Arias vitella and other caterpillars. Okay, it is effective against caterpillars. Next we have Tilonomus rimus. This is egg parasitoid. So from this you have to remember that Trichogramma is what kind of parasitoid? Whether it is egg parasitoid, whether it is larval parasitoid. Okay. So in this you have to remember this part. And you should know each and every parasitoid and predator, that it is effective against which of the pest or what is its target pest, and also which of the parasitoid it is, like it is egg parasitoid or larval parasitoid. Is it clear? This is the important question they can ask. Again, the Johnston's organ is primarily associated with Johnston's organ. Johnston's organ is primarily associated with which? Antenna. Yes, antenna is correct. Very good. Antenna. So, Johnston's organ is associated with antenna. And what is the purpose of antenna? They are hearing, detection and vibration. Okay, flight orientation. So, these are the major role of this organ. So what about mouth part? क्या है? What are the mouth part? Mandibles, maxilla, labium. So these are the mouth parts. What about antenna? Specific in antenna, that is Johnston's organ. That is important in antenna. Johnston organ that is present in the antenna, and it is important for hearing, detection of what? Detection of vibration, flight orientation, and sensing antennal movement. So direct a question can be asked that Johnston's organs is present in which? That is antenna. Okay. What is this cerci? That is a paired abdominal appendages. Okay. What are they? These are the paired abdominal appendages. These detect the air currents. Okay. So these are the different organ or specific organ. These two. That cerci and the Johnston's organs. Is it clear? And where exactly it is located on the antenna? That is, it is located in the pedicel or second segment of antenna. Okay, Johnston's organ exactly located at the second segment of the antenna, that is pedicel. Yes, very good. This is correct. Is it clear now? Next we have, which of the following insect transmits sandal spike disease? Firstly, you will discuss what is the causal organism of sandal spike disease. This is very important. What is the causal organism of sandal spike disease? Virus, bacteria, what is it? Anyone? Phytoplasma. Very good. Phytoplasma is the correct answer. This is again important question. They can directly ask that sandal spike disease is transmitted which of the following? Virus, bacteria, fungus, or phytoplasma. So sandal spike is transmitted by the phytoplasma, and this phytoplasma is transmitted by which of the insect vector? That is Riderator bimaculatus. Okay. So next we will discuss what is it? Common name, it is a leaf hopper, or they can directly ask that sandal spike disease is transmitted by aphid, thrips, white fly, or leaf hopper. So answer will be leaf hopper. Correct? Next is, this is also important, rice tungro virus. Rice tungro virus is transmitted by green leaf hopper, which is Nephotetics virus, as we have discussed in the last question also. This is important. And vector of Vigomo virus is also important. Vigomo viruses is the group of viruses, okay, that is transmitted by Bemisia tabeci, which is the white fly. So from here you have to remember two questions. One is rice tungro virus, vector of rice tungro virus, that is Nephotetics viruses, green leaf hopper. And what is the vector of Bigomo viruses? That is white fly, that is Bemisia tabeci. So this is the important one from here. Next we have, which of the following insect growth regulator act as a chitin synthesis inhibitor? This is very important and commonly asked question about this chitin synthesis inhibitor in insect. Which of the following? So answer will be diflubenzuron. Okay. So it is a chitin synthesis inhibitor. These are the example. One more is JH mimics. What are JH mimics? Juvenile. Yes, Nila Parvata lugens is brown plant hopper. Very good. JH hormone analog. What are their role? They maintain immature stage and prevent metamorphosis. They can also ask that what is the role of JH mimics or juvenile hormone analog. So what is their role? They maintain their immature stages. Okay, prevent metamorphosis. Clear? And what is the role of this chitin synthesis inhibitor? They prevent the formation of insect chitin. Is it clear? Insect cuticle during the molting. So again, these are important questions. Next is ecdysone and agonist. Okay, so they also cause molting hormone. This ecdysone, what is it? It is a molting hormone. What is ecdysone? This is molting hormone. So again, they what they are causing? They are causing incomplete molting. So you have to remember the examples of these. Example of what are associated with these. Okay. Next we have Indian species of lac insect commercially exploited for lac production. Which of the following? Again, this is very important, that which of the lac insect is exploited for lac production. So Caria laca is the correct answer. Which is Indian lac insect. Okay, which is Indian lac insect, that is a commercial source of natural lac. What about this Dactylopius coccus? This is a cochineal insect. This is also very important because it produces cochineal dye, that is carmine, from which Apantia. But here important is this Dactylopius coccus, its commercial importance is to produce a cochineal dye. It is producing a cochineal dye. Next we have Ericeros pella, that is Chinese white wax scale. So it produces white wax. So that's why these insects are important. It produces white wax. Next we have Ceroplastus rubens. What is it? What is it? Sandal spike we have discussed, that is a leaf hopper. Okay, so what about this Ceroplastus rubens? This is red wax scale. That is what? Pest produces wax. Okay, so again these are important thing, that what is the role of cochineal insect, that carmine dye is extracted from which this cochineal insect. What about Chinese white wax is extracted from this white scale, and red wax scale is producing wax. Okay, so these are the commercially important insects, these were. Next we have, which of the following stored grain pest as a external feeder? Which of the following is external feeder? See, very good. This is Tribolium castanum. This is the external feeder, which is red floor beetle. Okay, these are the external feeder. So here question can be asked directly, that which among the following is external and internal feeder. Okay, so what about rice weevil? It is also feeding internally. Okay, what about lesser grain borer? It is also feeding internally. Red floor beetle we have discussed, it is external feeder. Specifically external feeder. What about pulse beetle? It is internal feeder. So among them, red floor beetle is the external feeder and rest of them are internal feeders. Is it clear? So again, this topic is important. They can ask that which among the following is external feeder and which among the following is external or internal feeder. Is it clear? Okay. Next we have wax gland responsible for V-wax secretion in worker honeybee are located on. Again, this is basic question, but you should know that wax gland, where they are present. So wax gland are present in the ventral side of the abdomen. Very good. This is the correct answer. V is the correct answer, that it is present in the ventral side of the abdomen. So what about this wax gland? What is their role? We just learned that secretion of the V-wax. Okay. And what about its lateral side of head में क्या है? Compound eyes and antenna. And dorsal side of abdomen, that is tergites. And dorsal side of thorax we have thoracic dorsum. But here this is only important, that wax gland which are important, they are present in which side? So they are present in the ventral side of the abdomen. So this is important here. Next we have, which of the following parasitoid is commonly used for biological control of rice leaf folder? Again, this is very important question can be asked from rice leaf folder, that what is the parasitoid that is commonly used in the, anyone? B, no, it is trichogramma, but it is Trichogramma japonicum. Trichogramma is correct answer, but it is Trichogramma japonicum. That is a egg, of course, Trichogramma is egg parasitoid. This is also very important that this Trichogramma is what? Egg parasitoid. It is egg parasitoid. And what about this Encarsia formosa? This is white fly. Okay, Encarsia formosa is white fly parasitoid and it is a nymphal parasitoid. And what about Bracon brevicornis? We have discussed it is a larval parasitoid. So here you have to remember this specifically, that which of the following parasitoid is whether egg, larval, nymphal or what kind of parasitoid it is, and also what are the major target pest of these parasitoids. Is it clear? Next we have insecticides. Spinosyd primarily act on, again, mode of action of these insecticides. So it work on nicotinic acetylcholine receptor, which are generally responsible for the proper functioning of the nervous system. So they are acting on these receptor, nicotinic acetylcholine receptors. Okay, so I have written all the common chemical groups here and their insecticide and their primary mode of action along with their examples. This table is very important. You just go through this and remember all the insecticide group, what is their mode of action and their examples. Is it clear? Next we have, which of the following insect has piercing and sucking type of mouth part both in nymphal and adult stages? Piercing and sucking type of mouth part is present in which of the following? Anyone? Yes, this is white fly. Vemisia tabesae is a white fly and it contains piercing and sucking in both nymph and adult. So here I have given a list of all the mouth parts present in this and different type of metamorphosis along with its major host crop. Now there are different mouth part like chewing and biting type of mouth part, piercing and sucking type, siphoning, sponging, chewing and lapping, rasping and sucking type. So here you have to remember all the mouth parts. Okay, different mouth parts present in the different insects. Okay, so this topic is very important. Is it clear? Next we have the pheromone commonly associated with the queen honeybee is. Which of the pheromone that is present with the queen honeybee? Which of the following? So it is 9-oxodec-2-ionic acid. So this is the pheromone that is associated with the queen honeybee. Okay. So what is the role of this? It is also known as queen mandibular pheromone. And what is the role? It attracts drones during mating, maintain colony cohesion, okay, and suppress worker ovary development and queen cell formation. This is the role of this, what? Queen pheromone. Is it clear? Okay, next we have which insect pest cause characteristic dead heart and white ear symptom in rice? Again, this is important. That is dead hearts and white ear symptom. These are the typical symptoms associated with the crop. So, what are the different insect pests? No, this is yellow stem borer. Yellow stem borer produce typically dead heart and white ear or white head symptoms in the what? Crop. Okay, what about the plant hopper? Hopper burn is the typical symptom of the brown plant hopper. Is it clear? And what about leaf folder? As its name suggesting, Leaves fold longitudinally and larvae scrape the green tissues. Is it clear? Okay. Next we have the larval stage of, yes, again this is very important, larval stage of blister beetle that actively search for host is. What is the larval stage of blister beetle? That searches for the food or that searches for the host. Which one is? Planidium, triungulin, compodiform larva or scarabiform larva? Which of the following? So, this is triungulin instar, or triungulin is the first larval instar. This can be asked that triungulin is the what is triungulin? So it is the first larval instar of blister beetle. Is it clear? And white grub is scabiform, which is C-shaped. White grub is C-shaped, which is called as the scabiform. And in blister beetle, there is important metamorphosis. That is, what is it? Metamorphosis, hypermetamorphosis. Is it clear? That is present in the blister beetle. Okay. Next we have, which of the following insect is the primary pest of stored grain? Now what is primary pest? What is the secondary pest? So which of primary pest is Cytophilus or Ize? So what is primary pest? So primary pest, that is directly feeding on the grains, okay, directly feeding on the grains. And what are the secondary? That feed on that broken part or broken grains, cereals or processed food. That are the secondary pest. Okay, generally secondary pests are the external feeders. This is also important. These are the external feeders. Okay. Feed on grain directly, yes, these are the primary, and feed on the dead or broken, that are the secondary. Very good, this is the correct one. Next we have the predatory beetle, Cryptolaemus montroseri is mainly used against. Cryptolaemus montroseri is used against which of the following? Predatory beetle. It is a predatory beetle. Very good. Mealybugs is the correct answer. So it is used against the mealybug. Target pest is mealybug. Clear? Next we have which type of diapause. This is again important topic that occur during the egg stage of silkworm, that is Bombyx mori. Which type of diapause? D, very good. Embryonal diapause is present in this. And what are the different imaginal diapause occur in which stage? In adult stage. Okay. What about this larval diapause? Larval stage, pupal diapause, pupal stage, and embryonic in the egg stage. What is diapause? We all know that it is a resting stage or surviving stage during the unfavorable conditions. Okay. During the unfavorable condition, it is the surviving stage. Due to unfavorable conditions. Is it clear? Next we have, which of the following insect order possess haustellate mouth parts and scaled wings? Haustellate, or we can say, what is this haustellate or siphoning? Which we having the proboscis. So it is present in butterfly, which is in the Lepidoptera. Very good. C is the correct answer. Lepidoptera is the correct answer. So haustellate type of mouth part. Mouth parts, you have to study mouth part, different mouth parts and their examples. Okay. So I have written here also, butterflies and moth generally process siphoning and haustellate mouth parts, and these mouth parts form long coiled proboscis used mainly from sucking nectar and other liquid foods. Is it clear? Okay. Next we have, which of the following is the principal vector of little leaf disease of brinjal? Firstly, you will tell me little leaf of brinjal is again caused by what? Little leaf of brinjal. Yes, this is correct. Hishimonas ficatis is the correct answer. But tell me, little leaf of brinjal is caused by what? Butterfly's name in Kannada is chitte. Okay. Okay. So what is this? Yes, very good. This is also important. That is phytoplasma. This is very important. Little leaf of brinjal disease is caused by phytoplasma, and that phytoplasma is transmitted by Hishimonas ficatis. Next we have, which of the following natural enemy is a larval pupal parasitoid of Helicoverpa armizera? This is again very important question, that larval pupal parasitoid of Helicoverpa armizera. Okay, so answer is Campoletis choleridae is an important parasitoid of Helicoverpa armizera. Is it clear? Trichogramma, you should know it is an egg parasitoid, and Carcia formosa is also a parasitoid. Is it clear? Yes, very good. C is the correct answer. Again, this is important organ. Firstly, we have discussed Johnston's organ, that is associated with antenna. Okay. Next, we have tympanal organ in insect primarily function as tympanum organ. Everyone know tympanum related to ear. So it is associated with the sound perception. So tympanal organ is an auditory or hearing organ found in the several insects. Okay, it consists of thin membrane called, that is tympanum. Okay, so sound perception is the correct answer. So we discussed two organs, that is Johnston's organs, that is associated with antenna. Next is tympanal organ, that is associated with the hearing perception or sound perception. Is it clear? So this was all about today's session. Hope you enjoyed the session, and you should go through all the topics we have discussed regarding pesticide, insecticide, all the larval parasitoid, predators, everything. Okay, so this was all about today. Thank you very much.